Greetings and welcome to the 5th Origins Institute talk for the 2020-2021 academic year. My name is John Stone, or Doc Rock. I'm the director for the Origins Institute and your host. We are recording the proceedings and will make the video available for sharing. We will hold a brief question and answer period after the talk. Please input your question into the Q&A chat box, and we will address as many questions during the Q&A period as time allows. Before the introduction, I'd like to relay some information about the Institute. The Origins Institute is one among 5 research centers and institutes in the Faculty of Science and 70 in total at McMaster University. These centers and institutes discover solutions to complicated problems by bringing together interdisciplinary, multidisciplinary, and in our case, transdisciplinary teams. These teams collaborate with academic, government, and industry partners, and you. We are collaborating at this very moment. Community outreach and engagement is a priority for all centers and institutes at McMaster University, which is why we host talks in collaboration with the McMaster Alumni Association. We continue to showcase the Origins Institute mandate with our talks this year, combining our prime research directive, astrobiology, with original institute themes. Our guest today is an evolutionary biologist who therefore could address the species and biodiversity theme, but in parallel analog with the origin of species, we'll concentrate today on the origin of life. Dr. Antonio Lazcano is a professor in the School of Sciences at the National Autonomous University of Mexico, Mexico City, and a world authority in origins of life research. Professor Lazcano has been a visiting scholar and researcher all over planet Earth, including the Scripps Institute of Oceanography. Among the most remarkable credentials earned is an illustrious career. Professor Lazcano was president for the International Society for the Study of the Origin of Life, thereby bonding with names like Alexander Oparin, Stanley Miller, and Sandra Pizzarello. We are privileged, honored, and happy to have Professor Lazcano, who will use viruses and viroids as subjects to deliver an infectious talk about the currently widely held hypothesis for the origins of life, the RNA world. Professor Lazcano.
Thank you so much. I'm absolutely delighted to be with you today. I have to say that well over a year ago, I was invited by my friend Paul Hicks to give a seminar in McMaster University. And then, of course, the pandemic began to develop and we decided to cancel it. But now, thanks to all these media that we have available, the show must go on, can go on. And it's really quite a pleasure and an honor for me to be able to talk about my work. I have to say that I'm not an infectologist, I'm not a physician, I'm not an epidemiologist. I'm a biologist who has been very, very keen on the origins of life for all my life. And but when the pandemic started, in my lab, we realized that we had a very powerful intellectual framework to understand the appearance of a new virus, namely Darwinian theory, evolutionary theory, and that because of our work with the possibility of having an RNA world in the very earliest stages of evolution, we did have also the intellectual understanding of the genetics, the structure, the evolution of RNA systems. So then let us say that armed with all the instruments for bioinformatics, we decided to delve into the origin and evolution of the SARS-CoV-2 virus. And then we realized that this just fitted perfectly with our own interests in the origins of life, in the RNA world in particular. So that is the reason why the title of my talk is precisely the RNA world, insights from the RNA virosphere. And what I would like to share with you today is how we can actually look at the RNA viruses to get some insights, to have some idea of what the RNA world was soon after the origins of life on Earth. Now, let me start by saying that 1952 was quite an extraordinary year by all accounts in scientific terms. It was a year in which Watson and Crick published a double helix model of DNA in a short paper published in Nature in April 1953. And then a couple of weeks later, Stanley Miller, who was then a graduate student at the University of Chicago, working with Harold Urey, demonstrated demonstrated that if you simulate the conditions of the primitive Earth, you can actually synthesize in a very rapid time, in a very quick interval of time, you can synthesize amino acids, hydroxy acids, and so on, thus proving that in principle, you could have those organic molecules in the primitive Earth prior to the origins of life. Now, what I think is absolutely essential to keep in mind is that the proposal of the double helix model of DNA really changed biology. I think it really changed all of science, but really changed life sciences in a way that now if you want to have a theory about the origins of life, it really has to be consistent with what molecular biology was teaching us. So clearly the emphasis was to understand how the components of DNA arose, how the protein synthesis took place, and so on. And this really plays a very strong demand on the studies of the origins of life after that year. Now, what is absolutely amazing is that just 1 year after these two fantastic papers were published, the group of very, very distinguished scientists, mostly driven by the energy of George Gamow and James Watson, decided to create the so-called RNA Tie Club in 1954. The RNA Tie Club was created in the Cosmos Club in Washington, for mostly because the whiskey was supposed to be very good and a very low price there. But when you look at the list of the members that were part of the RNA Tie Club, one is absolutely overwhelmed by the quality of the science they produce. And their main concern, even if the ties were not pretty, but their main concern was really to try to understand the role of RNA in protein synthesis and general in biology. And ever since, I have to say that RNA began to gain most prominent role in biology. And it's fair to say that from the early 1950s onwards, the road to what we call now the RNA world was paved by the embracement of the idea that primitive life had RNA genomes, and it was proposed, as far as I know, first by John B.S. Haldane, who was a towering figure in biology, by John Di Bernal, also a major, major figure in crystallography and in science in general, John Piri, Oparin, Beloselski, and so on. So the people began playing with the idea that the primitive genomes were actually composed of RNA. Secondly, during those times, the people were a number of people with an extraordinary insight became fully aware that if you want to have enzyme catalytic activity, you require coenzymes. And most of the coenzymes were actually ribonucleotide derivatives. They really, people like Beloserski, who was a Soviet, Russian, Aikin, Handler, or Gail White in the USA, really were in the context of evolutionary biochemistry. One has to realize that since the late 1920s, 1930s, the people were looking at biochemistry as a unified field that could actually be developed to have a sort of universal rules that could apply to all cell, to all living entities. And of course, they were very much involved with an evolutionary perspective. So evolutionary biochemistry started to develop. And in this, and within this context, is that the people began saying, what if prior to proteins, what we had were metabolic pathways catalyzed by ribonucleotide coenzymes. And finally, in the late 60s or in the 1960s, people like Smith, like Francis Crick, Leslie Orgel, became very, very much aware that RNA, like proteins and unlike DNA, has very complex, can adopt very complex three-dimensional structures. And this led them to propose the idea that indeed RNA could be catalytic. And I think that all this culminated in 1968 when Francis Crick and Leslie Orgel published a paper in the Journal of Molecular Biology suggesting what we call now the RNA world hypothesis, namely that prior to the evolution of DNA, prior to the appearance of enzymes, the primitive biological entities in the earth were actually having RNA as genome as a genetic polymer, and that these primitive RNA molecules could replicate via catalytic RNAs that they were encoding. So one could say that the RNA world hypothesis is the outcome of biological intuition, of an evolutionary framework, and of a very deep understanding of what molecular biology is. Very soon, the RNA world began to be connected with the idea of prebiotic chemistry. And if one looks at the history, a history that starts in the 19th century with the classical synthesis of urea, alanine, sugars, and so on, one can see how in the 1920s, a number of theoreticians, mainly Oparin, Haldane, Lippmann, Harvey, but above all, Oparin, came to the idea that these chemical syntheses were an indication that equivalent processes could have taken place in the primitive Earth. This led to the idea of a heterotrophic origins of life. These ideas were strongly supported by Stanley Miller 1953 experiments. And I have to say that Stanley Miller was at the time about the age of most of the graduate students that are kindly attending this seminar. Then in 1960, 1961, John Norrow was able to synthesize adenine. So you could demonstrate that in the primitive conditions, prebiotic chemistry could lead to the formation of some of the components of DNA and RNA. And from 1968 onwards, we have the RNA world concept. Now, there are different ways of representing the RNA world hypothesis, but this is a highly simplistic, but that I found extremely useful version of what will be the RNA world. We can synthesize, we can understand the accumulation of the primitive soup in the early Earth without any major problem. We recognize the key role of RNA in many biological activities. We really do not know, and I have to underline this, how we went from the primitive soup into the RNA world itself. We believe that proteins are actually the outcome of evolution of an RNA world interacting with amino acids, with oligopeptides, and so on. And in this scheme, DNA is a latecomer that evolved and was and led to more complex genomes. Now, this is of course a hypothetical scheme, but I will try to argue that all the evidence that we have, in fact, support this in general, this scheme I'm presenting here. What is the best example that we have of catalytic RNA and that indeed protein biosynthesis emerged in an RNA world? Well, we have the demonstration by crystallography, and this scheme comes from the work of Lauren Williams and his friends, my colleagues at Georgia Tech, that if you look at the catalytic side of the large subunit of the ribosome, you don't have any proteins in the actual place where the peptide bond is being synthesized. So the studies of people like Ada Jonath and Tom Stites and many others have actually led to a very, very clear demonstration that indeed the ribosome, the catalytic side of the ribosome, requires no protein, supporting hence the idea that indeed a protein biosynthesis started in an RNA world thanks to the interaction of RNA with a huge number of additional molecules that must have been present in the primitive Earth. Now, what could be understood as the RNA world? I think that there are many definitions of the RNA world, including some that are contradictory ones. This is not surprising. It happens in science all the time. But I think it's fair to say that it was an early, perhaps primordial, perhaps on the transition from chemistry to living beings, a stage during which RNA molecules and ribonucleotides played a much more conspicuous role in heredity and metabolism. And I have a scheme that actually tries to summarize this idea. If you want to have RNA, you know that you need ribonucleotides. The ribonucleotides are synthesized via what is essentially the same pathway, biochemical pathway, metabolic pathway in all living entities that we have studied so far. And of course, once you polymerize ribonucleotides via an RNA polymerase, we have RNA. And in fact, we can divide RNA into major groups. We can have coding RNA that can include riboswitches, as in the messenger RNA, or as in the case of the RNA genome that we see in the coronavirus, in famous coronaviruses, or we can have non-coding RNA, and this in turn can be divided into small non-coding RNAs, like the microRNAs, like the RNAs associated with the PIWI system, which play essentially a regulatory role, or we can have large non-coding RNAs, like the ribosomal RNA, the transfer RNA, the primer RNA, and the large non-coding RNA, which actually play also a key role in regulation. If we want to have a DNA, we need desoxyribonucleotides, and in absolutely all living entities, if you want to have the oxyribonucleotides, biochemistry needs to reduce very rapidly, needs to reduce ribonucleotides to form desoxyribonucleotides. So this is a very strong biochemical argument to assume that indeed RNA, or at the very least ribonucleotides, came prior to the oxyribonucleotides. And finally, one thing that we have been working in my lab for some time is the study of the biosynthesis of coenzymes, histidine, other hormones like cyclic AMP, for instance, or some antibiotics are actually derivatives of ribonucleotides. So you see that many of the key catalytic and structural roles of RNA and ribonucleotides in extant biology may be considered as relics of the RNA world. Now, the only systems that we know of as today that actually use RNA molecules as genetic material are viroids, of course, and RNA viruses. And there is a fascinating quote that I always like to mention that was made by Holland almost 30 years ago in a lovely paper that he published in this book, in which he says that today's biosphere is DNA-based, and the only known RNA life forms are RNA viruses. And he puts life forms between quotation marks, which I think is a very fair description of the issue of whether they are alive or not. These depend on DNA host cells for their existence. Whether any existent RNA virus genomes contain sequences, vestiges of the earliest RNA life form is unknown and probably unknowable, but it is clear that RNA viruses are ubiquitous, extremely successful cellular parasites of considerable disease importance. And of course, the pandemic times we are living is a very clear demonstration of how right Holland was in writing this. Now, the question that one can keep asking all the time is, are extant RNA viruses good working models of the RNA protein world evolutionary stage? Well, let us look first at RNA viruses. It's very difficult to come up with a reasonable taxonomy systematic of viruses. There are many arguments going on, among other things, because we are using the taxonomy developed for microbes, for bacteria, to try to classify virus properly. But in 1971, David Baltimore decided it was good to recognize that viruses can be divided into two major groups. Viruses made of DNA, which can have a single-stranded DNA or double-stranded DNA genome, and RNA viruses. And you can see in RNA viruses that we can have single-stranded positive RNA, which is really used as messenger RNA. We can have single-stranded negative RNA, which needs to be transcribed to become a single-stranded positive RNA and then be translated, double-stranded RNA, and retroviruses like the infamous case of the HIV virus. Well, one thing that I would like to say is that this is a very convenient way of classifying, of dividing RNA viruses. But as time goes by, I have to say that I'm convinced that indeed RNA viruses have such an extraordinary diversity that one could compare them, for instance, with the animal kingdoms. The animal kingdom includes corals, includes sponges, but it also includes elephants. And you see the extraordinary diversity of animals that you have between the frontiers of this particular kingdom. Well, I think something comparable is true about RNA viruses. Now, I think it's very unlikely that RNA viruses are descendants of primitive RNA-based life forms. One argument that I think can be used to support this contention is the fact that with very few exceptions, like the PQ beta virus, RNA viruses really mostly are parasites of eukaryotes, and eukaryotes are, of course, latecomers in the evolutionary history of the biosphere. However, the evolutionary analysis of their viral replication strategies, of their genome sizes and organization, of their mutation rates and gene activation processes may provide significant insights into the structure of evolution and evolution of early cellular genomes prior to the emergence of DNA genomes. So let me just clarify this point. I will never assume during this lecture that RNA viruses are direct descendants of primitive RNA life forms. I think they are very good working models, and as a model, they can have a number of strong limitations. Now, due to the lack of proofreading and repair mechanisms, RNA viral genomes have an error-prone replication. They have very, very high mutation rates. Don't forget that one thing is to have high mutation rates. Another one thing is to have high evolutionary rates. They have typically small sizes. They have extensive genetic variation. They have a different genome organization strategies, as I have already mentioned, and they are limited by a number of adaptive constraints that we can recognize without any problem. So, well, the first thing that one would like to understand is how do RNA genomes replicate? And of course, we know that all DNAs, all RNAs replicate, I mean, any nucleic acid replicates via a polymerase. And for many, many decades, the issue was how can we come up with a reasonable model of an RNA-dependent RNA polymerase? And one of the papers that I found most insightful was published by friends and colleagues from the Pasteur Institute in Paris by Marc Delarue et al. In this paper published actually 30 years ago. And here you have an attempt, it was a very important paper, an attempt they made to compare the sequences of different RNA polymerases of different RNA viruses. And you can see, comparing every line here, how very little conservation we have. And this is just an indication of how rapidly the information in sequences can be eroded in an RNA system. Now, this is true for sequences, is not true at all for three-dimensional structures. We know that many polymerases actually have what we call the right-hand model. They have the so-called fingers, they have the thumb, and they have the palm domain. And these are traits that we can distinguish without any problem in absolutely all RNA-dependent RNA polymerases and in a number of DNA polymerases, as we can see. And I'm comparing it here with the Rodin's model of a head. And the thumb will be here, the fingers are here, and these interact with the template and the substrate. And the oldest, the most conserved both in sequence and in a 3D structure is precisely the palm domain, which is the one involved in the formation of the phosphodiester bone. Now, if one looks at the structures, the available structures of RNA-dependent RNA polymerases, RNA-dependent DNA polymerases, as in the case of reverse transcriptase, or most monomeric DNA-dependent DNA polymerases, one can see that the three-dimensional structure is very highly conserved. We see this so-called the right-hand model. And of course, again, the sequences are not so conserved, but the crystal structure really provides us with an extraordinary information. Well, several years ago, three groups independently proposed a way to classify, to study the phylogenetic relationship between these monomeric polymerases using a phylogeny, developing a phylogeny based on the three-dimensional structures. And we were one of those groups. And in 2050, we published a molecular phylogeny grouping and showing the evolutionary relationship between these polymerases. And here you have, for instance, the DNA pol B, which is a monomeric DNA polymerases. It reads DNA, forms DNA, and it's really the eukaryotic replicative polymerase. You can see here the DNA pol that we see in, for instance, in mitochondria. And here you have, by the way, here the branch that connects the DNA polymerase of the T7 phage, as well as the the RNA polymerase of also the same fate. These are DNA-dependent DNA polymerases. And here you have a number of viruses, the polymerases of a number of viruses, including reverse transcriptases. So this is really what this phylogeny is really telling us, is that, first of all, again, all these monomeric polymerases have a monophyletic origin. All of them are evolutionarily related, and they can easily switch in the course of evolutionary time. They can easily switch substrate and template without any major problem. And indeed, we can see that in the lab. Without any problem, we can force that DNA polymerase fragment without the exonuclease domain. We can force it to read RNA or DNA. We can force it to use ribonucleotides as substrates. And then, of course, with reverse transcriptase, we know exactly what is going on. One thing that you know, of course, is that reverse transcriptase, when it's replicating the RNA of the HI virus, introduces a huge number of mistakes. We know that if we cut off the exonuclease domain of DNA pol, the same will be true. It will introduce a high number of mutations, point mutations, and this can be understood without any problem. In this graph, we put the size of the genomes on the x-axis and the mutation rate on the y-axis. You can see how eukaryotes and bacteria, we have very large genomes, and we have a really low amount of mutations. This is perfectly easy to understand. It's clear that we have repair mechanisms, we have editing mechanisms, and so on. And because of the large genome, we can encode for all these molecules. If we look at double-stranded DNA viruses, then of course the mutation rate is higher because the size of the genome is smaller, and they do have some repair and some editing mechanisms. Sometimes they use those of the cell, as in the case of the smallpox virus, but in general, their mutation rate is higher than that of eukaryotes and bacteria, and certainly lower than single-stranded DNA viruses or RNA viruses. Why do we have this graph that is so clear here with viroids over here? I'm not going to address their biological nature today. Well, for a very simple reason. If we have too many mutations, the genetic identity of the polymer is low. If we have very low level of mutations, that you don't have diversity, and if you don't have genetic diversity, you lose the possibility of the population responding properly to the selection pressures. Now, because of the absence of editing and repair mechanisms, RNA viral genomes tend to have very small sizes, from 2 to about 33 kilobase pairs. They have a limited coding capacities, and this has led to a number of fascinating evolutionary strategies. For instance, to cope with these small sizes, some viruses use what I call the matryoshka strategy. All of you have seen these Russian dolls, and what you can find, for instance, in some retroviruses is the overlapping genes. So without enhancing the size of the genome, you can actually be encoding in the same sequence for more than one protein, which is a very reasonable strategy equivalent to the matryoshka dolls. Other viruses actually overcome what we call the eigenlimit, this idea of losing the genetic identity if the genome is too great and accumulates too many mutations, for instance, by having segmented RNA genomes, which is what we see in the case of influenza. You know that we need to have vaccines ready every year when the new season of influenza comes, because of course we have this exchange of pieces of RNA in what is a fragmented genome. And one of the most fascinating cases of segmented RNA genomes, I think, is in the case of genome viruses, in which the virus is not one of these capsids, but all of them at the same time. And if you want to have a proper infection, you actually need that the vector actually infects the host with all those capsids at the same time. This may look very strange to us, but remember that we ourselves have this kind of genomes. We have a very significant amount of DNA in our nuclei, but we also have essential genes in our mitochondria. So this strategy, this evolutionary strategy in which one can recognize the division of genomes in more than one component is very simple. Now, I just say that because of this absence of editing and repair mechanisms, RNA viral genomes tend to be very small, but this is not true for coronaviruses. The only known example of editing mechanisms in RNA viral systems are precisely coronaviruses, including the SARS-CoV-2 that is causing so much harm now. They encode for a 3', 5' exonuclease, which I will refer to as ExoN, which has led to a rather significant increase in the size of their genomes compared to other RNA viruses. And there is a fascinating paper that I strongly recommend to you all that was published about 15 years ago by the group of Gorbalenia et al. In virus research. And here you see in the x-axis, you see the sizes in ribonucleotides of different RNA genomes. Here you see the different RNA genomes, and you see that most of the RNA viruses, as I have already mentioned, have relatively small genomes, something between 3,000 to about 12,000 or 16,000 at the most. But when you look at coronaviruses, for instance, SARS-CoV-2, you see a genome that is almost 30,000 ribonucleotides in length. You see truly an enormous, a huge gap between the largest RNA viral genomes that belong to, they are part of viruses that have no editing activity. And in the case of coronaviruses, clearly the difference is very significant. Well, we have a huge number of sequences of coronaviruses, and one can analyze, one can compare the sequences of the exonuclease domain. And here you have the phylogeny of the coronaviruses exonuclease. You see here for different coronaviruses how highly conserved they are. They have a conservation, sequence conservation of about 93, 94%, which is huge. And you see how very rapidly they have expanded. This is part of a paper that will be published this coming Wednesday that comes from my group. And here you see this very rapid expansion of the sequences evolution. Now, one can not only look at the sequences, but one can also look at the three-dimensional structure. And I just mentioned, of course, everybody knows this, that polymerases are the essential enzymes in nucleic acid replication. Here you have the, oh, sorry, it's in Spanish, but I think you can cope with it. You have the 3D structure of the RNA polymerase of hepatitis C virus. Again, you see the thumb, the fingers, and the palm domain. And here you have the DNA polymerase of E. coli, number 1. And here you have again the fingers. I'm sorry, it's in different colors from here. Fingers here, it's in this pinkish kind of color in the RNA polymerase. You have the thumb in blue, also in blue here. The palm domain in yellow, the palm domain in green, and you see here the extra domain that belongs to the exonuclease, the 3', 5' exonuclease. How does this exonuclease work? Well, we have a very, very good understanding of the way it works. If you have DNA that is being replicating, you see the DNA polymerase that goes on and forms a phosphodiester backbone between the nucleotides without any problem. But here it makes a mistake. If you have C, what you should have facing it will be a guanine and not adenine. So the polymerase made a mistake. Well, very rapidly the exonuclease comes into action, breaks the phosphodiester backbone. The phosphodiester backbone is already strained because you don't have the localized electrons here, and this is cut off. Then it goes away to be eaten eventually by another polymerase, and then the polymerase goes on replicating the DNA without any problem. Well, we have a very, very good idea of the structure of these exonuclease domains, actually of the whole DNA polymerase. And here you have the different domains, the different components of the E. coli DNA poultry domain. And in red, you can see the exonuclease. And in August 2018, a rather bright, hardworking undergraduate joined my lab, Adrian Cruz Gonzalez, and he was working for his undergraduate thesis with Rodrigo Jacome, who is a friend, a colleague who developed the three-dimensional phylogeny of polymerases, and they decided to undertake the analysis of the exonuclease well before the coronavirus pandemic started. And one of the things they rapidly did by comparing the three-dimensional structure is to demonstrate that the DNA pol 3 exonuclease domain is structurally homologous to the exon domain, the exonuclease that we see in the SARS-CoV-2 virus. So this was very encouraging. Very rapidly, we started all comparing the exonuclease domain of the SARS-CoV-1. Now we can do it with the SARS-CoV-2 with the E. coli DNA pol exonuclease domain, and found that it's of course homologous to that we see in the fake T7, which is a DNA virus, DNA, the polymerase is DNA, of course, and with the Lassa virus, which is also an RNA domain. So you have here a homologous domain of about 300 amino acids that for exonucleases that is found in cells, in RNA viruses, in DNA viruses, and in the coronavirus, which is of course a DNA, an RNA virus. Well, we developed, as I already mentioned, this possibility of making very precise phylogenies based on 3D structures. And here is what will be published this coming Wednesday. Here you have that the double-stranded DNA viruses have taken the exonuclease from cells at least one time. The same is true for the RNA viruses, like the Lassa viruses, where it is just not really to edit the RNA that is being replicating, but mostly it affects an RNA molecule that is part of the host defense mechanisms. So here really it's used as a way of defending against the immunological system of the animals, including us. And finally, you have here another hijacking event that led to the robbery, if you wish, of the exonuclease domain by the coronavirus. So at least 3 times the exonuclease has been stolen, has been hijacked by viruses. So one could say that exonuclease genes have been hijacked by viruses at least 3 times by DNA fakes, P29, T4, T7, and by RNA viruses and coronaviruses, which have RNA genomes. In other words, both RNA and DNA are exonuclease substrates. In other words, like polymerases, exonucleases do not have a very strict substrate specificity. So when we thought about this, we think that it's reasonable to assume that the early evolution of exonuclease activity was a crucial event that could allow the size increase of primitive RNA genomes. We're extrapolating from the extant systems to the past. It may not be so difficult to evolve an exonuclease activity. After all, it involves a simple hydrolase reaction capable of destroying a phosphodiester bond in a polymer that's already strained due to a mismatch by base pair. And finally, that the postulated transition from small fragmented RNAs to much larger DNA cellular genomes would have been facilitated by the lack of absolute substrate specificities of exonuclease domains. You don't need to invent a whole new type of a protein. The protein that has already been developed in the RNA world could actually work work without any problems once you have DNA genomes. Now, this leads to the obvious question, which is, how did primitive cellular the RNA genomes increase their sizes? Because I already mentioned that with the exception of the coronaviruses, RNA viral genomes tend to be small, and the same was probably true for early cell RNA genomes. In other words, how do we come to these very complex genomes that we see today? Well, analysis of cellular genomes, which is something we can do without any problem now with the development of genomics, demonstrate the key role played by gene duplication events in their size increase. And let me put you a very good example. This is a paper published 30 years ago by Monica Riley and Bernard Lavedan at Orsay in Paris. Monica Riley unfortunately passed away a few years ago, and they did a phylogeny of the ATP binding protein of the ABC transporters, which play a key role in taking things into the cell or outside the cell, mostly into the cell. And here you can see the explosion of diversity, of substrate specificity of these particular proteins due simply to gene duplication events. Now, did gene duplication play a key role in the primitive RNA genomes? Again, let us look what RNA viruses are telling us. In RNA viral genomes, multi-gene families, which are the hallmark of gene duplication, are conspicuously absent, suggesting that gene duplications may occur, but their preservation is not favored. And then, of course, we already have the problem that I already mentioned several times, that the information contained in the sequences is very rapidly erode. So the high level of mutation rapidly erodes evolutionary information contained in sequences. Well, about a year ago, we decided to undertake the analysis of this issue, once again looking at three-dimensional structures and not at the sequences. And the paper is already published, not in the proper format, but you will see it in Virus Evolution, a paper by Alejandro Cisneros Martinez, Arturo Becerra, and myself. And so we decided to compare the three-dimensional structure of the genes that we found in a number of RNA viruses. And here you have the comparison of a fungal toxin of a virus that affects Ustilago, which is a Mexican delicacy, Huitlacoche we call it. And here you see how very clearly the three-dimensional structure is telling us that indeed there was a duplication of not an independent acquisition, but a duplication in the virus genome of a double-stranded RNA genome. There's another example that actually comes from the coronavirus, NSP3 protein, in which you can see here how very clearly one can see the duplication of a component of the SUD part of the NSP3 domains. And finally, you have here for the BP1, 2, and 3, which are components of the capsid of the VC retroviruses, which affect insects, are RNA positive, single-stranded RNA positive viruses. So here you have, without any problem, the indication that indeed there are the outcome of duplication. So in conclusion, I can say it's fair to say that extant, we believe this, that the extant monomeric polymerases like the ribosome RNA-dependent RNA polymerase, the A and B type DNA polymerases may be highly modified relics of an RNA-dependent RNA polymerase that appear prior to the development of DNA cellular genomes. Secondly, as suggested by the extant ExoN, the exonuclease domain, a key event in the evolution of genomes must have been the early emergence of an array of editing and repair activity, including the 3', 5' exonucleases. Actually, we think this was the first of all these to appear. And finally, the gene duplications and or recombination, which of course can also occur, may play a key role in the size increase of RNA cellular genomes very early in the history of the virosphere. So thank you very much. It's basically what I wanted to share with you, and I'm quite happy to answer questions or comments or remarks if feasible.
Thank you very much, Professor Lazcano. That was a beautifully complete talk, including history, science, with data and expertise, and we really appreciate it. One question was raised during your talk by a mycologist, a colleague who studies fungus, actually, about the graph that you showed with, I think, from Holmes' paper about the mutation rates.
Yes.
And the question was, are the mutation rates nucleotide mutation rates?
Yes. The mutation rates are measured here in the nucleotides. You can do the same, of course, with the phenotypic expression of those mutations. In those cases, the whole thing will be lower than. I think that the basic idea of having the nucleotide there was because you can have viroids, and viroids do not express any protein, so that fitted quite beautifully.
Thank you. And as you're aware, we have a very astute alumni audience as well, and they came up with some very interesting questions even before the talk, but I think they're more related now that you've given the talk. So the first question has to do with a type of RNA that doesn't receive much attention in the RNA world scenarios, and that is, what are the current hypotheses about tRNA?
About?
Transfer RNA.
Transfer RNA. I think it's a major issue. There was a paper published about a week ago suggesting that, in fact, ensembles of transfer RNAs can actually have a replicase activity. And I think that if you want to enhance the catalytic activity of RNAs, a way of doing so is adding additional chemical structures or components. And I think that's what really favored the selection of aminoacylation of transfer RNA. But I think it's a very, very open question. I have never been convinced by any of the proposals of an origin of transfer RNA. For the time being, I'm perfectly happy to see that the catalytic site of the ribosome has no proteins. But I completely agree with the person, with the colleague, with the student who raised the issue. I think it's a very, very key issue, because without it, you cannot understand the origin of the genetic code, of course.
Yeah. Another question, you did touch upon SARS-CoV-2 in your talk. And of course, that's on a lot of people's minds. And one of the Alumni Association members asked, kind of turning around your approach to the talk today, what implications, if any, does the RNA world hypothesis have for our understanding our world today?
Well, Aristotle said over 2,000 years ago, if you want to understand the nature of things, you have to understand the origin of things, which I think is quite true. I think that all the biochemistry that we have an enormous, a huge corpus of biochemical information of RNA can be applied to understand the genetics structure of the SARS-CoV-2 virus. For instance, we know since the classical works in the 1960s or so that the deamination of cytosine that will give you uracil is repair, of course, in DNA, that RNA viruses, well, RNA molecules don't have the repair systems against the oxidative deamination. You lose the amine, the ammonium group that you have, the NH2 group that you have in carbon 4 of cytidine. And now we have discovered that there are some mechanisms which favor that kind of mutation in the SARS-CoV-2 virus. We don't understand exactly why, but we know it's happening. So there you have a very clear example. The other thing that I'm very proud of this actually was the first paper published from my group during at the start of the epidemic, is that Rodrigo Jacome, who's now a colleague, he has a position in my lab, a permanent position as a full professor, began playing in the computer with the models that he had developed for monomeric RNA polymerases, for viral RNA polymerases. And we were preparing to close the lab really, that day we decided to close the lab. And then he called us and said, look, sofosbuvir, which is a medicament that is used to control the replication of hepatitis C virus, fits perfectly well in the RNA polymerase of SARS-CoV-1, the original, the cousin of the infamous SARS-CoV-2. And then he said, look, we actually saw by docking and so on, how the interactions of this medicament, this component with the catalytic site of the RNA polymerase. So we said, okay, that is telling you how highly conserved is a catalytic site of RNA polymerase. Let us propose that as a way of inhibiting the replication of coronaviruses. We sent the paper to Nature. It was accepted right away. I was surprised to see how rapidly it was accepted. We published it in scientific reports. And then about 2 weeks after the paper was published, a group of physicians, Rodrigo is a physician originally, but he found the right way in life, working in evolutionary biology. They contacted him from the Institute of Cardiology in Mexico, which is a very prestigious hospital down here. And they asked him, why don't we develop a protocol to try to see sofosbuvir in infected patients? So for me, it was a whole new world to discover, going from the bioethical issues to the design of medical protocols and so on. We were able to test sofosbuvir in 10 patients. No harm was done, which is in itself a fascinating result. And now we received the funds to do a larger essay. Now, what's fascinating is that in a space between like 2 weeks, when our paper was published, a colleague from Egypt came to the same conclusion. 2 groups, one in Austin, another one in New York City, and a group in Iran came exactly to the same idea, using different methods. So, I mean, that means that a lot of people were thinking in terms of how to inhibit a key component of the viral replication chemistry, but our approach was absolutely evolutionary.
Oh, yes, and let me see if I understand correctly. Your the clinicians contacted the former doctor scientist to, they took the action.
Right, absolutely. And for me, again, it's fascinating. I mean, all the controls you have to have, I mean, starting from the bioethical issues, I mean, you can always say no bacteria was harmed during this experiment, and no people from the animal rights groups will raise any issue against you. But when you talk about patients, I remember, I don't know the name of the patient, but which I shouldn't. But I remember that we were looking through all the list of patients that the clinicians provided. It turned out that one patient discovered that he or she, I'm not going to go into further details, was HIV positive. And then all the physicians say, no problem, let us look at the white blood, white cell count and so on. So for me, that was fascinating, but it also shows you the tragedies that can be involved in this kind of procedures.
Absolutely. Okay, one last question from our alumni. Is about genetic markers, and do you think that they played any roles in the history, perhaps in the RNA world, but perhaps en route to current world from the RNA world?
If by genetic markers you mean a sequence or a structure that can be used to follow the dispersal of a system that is replicating and evolving, my answer will be yes. The best marker that we have will be the catalytic site of the RNA polymerases, of the monomeric RNA polymerases. I hope I'm answering the question. I'm not quite sure there was the issue.
Yeah, it wasn't specified, so I think that's perfectly appropriate. And we have a second question from my colleague. I guess you can't keep a fungi down. Do positive and negative sense RNA viruses differ in their genome size and organization?
The organization is different, the sizes, it's the statistics are not so clear. I would say that in general, but I would easily accept a criticism if there are new data available. Positive RNA genomes tend to have a larger size than negative RNA genomes in general. But remember, and I love this question coming from a fungi specialist, remember that we really have a very strong bias set of databases. We know well about the RNA viruses that affect humans. We think we know a little bit, a reasonable amount of data about the RNA viruses affecting animals, especially farm animals. The veterinaries have done a wonderful work studying coronaviruses, among others, but we have no idea about the RNA viruses infecting amoeba or chytidiomycetes or diatoms. So we have a very human or animal-centered understanding of the biology of the viruses, and that is true for everything. We have in the front page of the newspapers a new epidemic affecting humans in, let us say, in Latin America, but we never see the same about an epidemic affecting amoeba.
Exactly, yes. Okay, thank you very, very much for an absolutely complete talk. Our Alumni Association thanks you, our students and our postdocs, our faculty and staff, all thank you very much for your time today.
Thank you, keep safe, and I do hope that, as we were talking just before the lecture, Mexicans look with so much sympathy Canada, and I hope the opposite is also true, that one should use these opportunities to enhance the academic exchanges and so on. And let us hope that with these new instruments that everybody's using, or when things come to return to some sort of normality, we can have exchange programs between our students or faculty members and so on. So thank you once again for the opportunity. And thanks, Paul, for being the first to promote this idea.
Thank you very much once again.